For the purposes of the recording, I'm David, and I'm just going to check that the recording really is on. Oh, of course not. Yes, okay, recording is on. Right, I'm David, I'm coordinator of the Maths Learning Centre, and this is my second lecture to the third year med students. Welcome to anyone listening who's not a third year med student, because I've directed you here because I thought you might find it helpful. And welcome to all of you faithful souls. Today I'm going to be, it's called Thinking About Data, but it's really about descriptive statistics. Because that's what descriptive statistics is a way that statisticians think about data. So in the first lecture, I talked about the four different major realms of statistics, where you've got descriptive statistics, which answers questions like what's going on, confidence intervals, which answer questions like what's this number, hypothesis tests, which answer questions like yes or no, and modelling and regression, which answers questions like what's the formula. And over the next few lectures I'll talk about each of those four areas, just so that you can have more information about them and therefore more power over them. So today I'm going to talk about how statisticians think about data and how they make graphs and summaries. And it's really important information because it will flesh out a little bit about how they make the decisions about what stats goes with what. So what I'm talking about here will help with that decision. All right, I'm just going to run over some of the things I said before. Statistics helps to answer questions. That's what it was originally designed to do. And the questions are about concepts. It might be about a single concept, such as how much potassium is there in a banana, or about the relationship between concepts, such as do men and women, does your gender affect your chances of participating in volunteer work? So those sorts of things. And most research questions that you want to answer with statistics are about the relationship between concepts. But statistics cannot answer questions about concepts. Okay? That's what an essay is for, talking about concepts generally. Concepts need to become variables before statistics can work on them. You need to measure them. You need to figure out how to measure that concept, and once you've measured it, you've written down the information about each individual that that is under study. They become variables, and I draw variables as nice, well-defined rectangles, whereas concepts are airy-fairy cloud thought bubbles. Okay, so it's not until your concept has been measured until it becomes a variable, and it's not until it's a variable that your statistics can deal with it. Okay, so for example, your classic example is depression. Depression's a concept. But you can't do statistics on it until you measure it. And you might measure it as mild, medium, severe. You might measure it as the sum of 20 Likert scale questions and add up that score, and then it would be a number. But once you've made the decision, then you can start doing statistics on it. And variables come in two types. There are two main flavours of variable. There is the numerical variable, and I tend to draw them with a box with the variable's name at the top and a symbol at the bottom to describe what kind of variable it is. So a numerical variable I describe with a nice little arrow with marks on it, which is the scale on which I measure my numbers. Numerical variables are actual proper numbers, and you can tell it's a proper number by how far apart the numbers are. That means something. So the difference between 1 and 2 is the same as the difference between 2 and 3. They have the same meaning. That's a real numerical variable. And it's also known as a quantitative variable, an interval variable, and a scale variable, depending on which textbook or resource you're looking at, or computer program. I call it numerical because that's what it's called in Medical Statistics at a Glance by Aviva Petrie and Carolyn Sabin. Very good book. You should look it up. There's about 5 copies in the library. So Medical Statistics at a Glance. And categorical variables are generally words for putting things in boxes, which is why I draw them as boxes. You're putting things into categories, and some people call them qualitative variables. So they're the two main types of variables. And last time we did this activity over here, where we had a heap of these different variables, and we had to decide what type of variable they were. So you may have one, you may want to take one as well if you want at some point, and I'll leave them on the table for you, and you can get them in a minute. And we decided with all of these variables what kind of variable they were and I'm just going to ask a couple of them now so that we can be sure that we're all on the same page. So I'm going to go to number 14. What country a person was born in? Is that categorical or numerical? Good. The change in the... No. What was I looking at? The change in the area of a melanoma. That one there. Yeah. When a mathematician says the change, they mean how far apart. So it's a proper number. Okay, good. That's 2, doing very well. And I do have something to say. How you choose to record your information, the way that you measure your concept changes what kind of variable it might be. So number 24, whether skin is hairy or smooth. What's that? And number 25, the number of hairs per square centimetre of skin. Numerical. They're both ways to measure hairiness. So if you wanted to talk about how hairy a person was, there are 2 ways to measure it and one's categorical, one's numerical, and that would make a difference to the sorts of statistics you can do. Right, that's the main point of that. I'll get back to the presentation. All right, so where have we up to? Concepts are about, questions are about concepts. Concepts need to be turned into variables by measuring them. And then the next stage is that subjects are sources of variables. So to a statistician, the word subject means whatever it is that is having information recorded about it today. So the things that are in your experiment or your observational study, they are the subjects. And to a statistician, people and bananas and petri dishes are just walking around with variables coming off them. And we measure the variables and then we use them to do statistics. Okay, so a person's not a person, it's just a source of variables. Okay, so my, I'm just going to check something. Yep, okay. So I've got a banana here. I could write down its age, which would be a numerical variable, its weight, which would be a numerical variable, what variety of banana it is, which would be a categorical variable, and the amount of potassium that I find in it, maybe per 100 grams, that would be a numerical variable. I might have a person and I might write down their temperature after having a meal with chili in it and their temperature after a meal without chili in it. Just continuing with my standard example. And I could have a petri dish, which is my subject, and maybe I'll write down the culture medium, what treatment I gave it, how many bacteria colonies there were on day 1, how many bacteria colonies there were on day 2, how many bacteria colonies there were on day 3. I can write any number of variables down about basically anything. My next point is that variables can be used to calculate other variables. So you don't have to measure the variable directly on the object for it to count as a variable statistically. So from my person there, I could calculate the difference in temperature between with and without chili. So I could go one minus the other, and then I'll get a new number, and that number still belongs to that person and still counts as a variable. And with my petri dish here, I could use those 3 numbers of colonies on day 1, 2, and 3 to calculate some sort of growth rate, you know, how many colonies it grows on average per day. And that's still, even though it's calculated on the individual, even though it's calculated on the variables, it still belongs to the petri dish, and so it still counts as a variable. So just because you calculate it doesn't mean it doesn't count as a variable. Okay. And I have to say this again, as I said last time, whenever I ask a statistician what I should tell students about statistics, this is what they say, is that information about the subjects goes into rows. So in this situation, I've got some people, and each person has a gender and an age, and whether I gave them chili and their temperature, and person number 1's information has gone in row 1, and person number 2's information has gone in row 2, and person number 3's information has gone in row 3, and so on. Now, this is not just information to keep the statisticians happy, because, you know, now that I've told you, I know it's full screen. Now that I've told you, I can say, good, done everything I need. It's not just that information. It's how a computer wants your data to be set up. Okay, it needs your data set up in a certain way in order to be able to do what it does with the data. And a very important thing that we need is that graphs will be built from this arrangement of data. And I'm going to come back to this several times before I finish today. All right, first kind of graph is a scatter plot. The subjects, or rows, are the points on the scatter plot. And information is represented by what the point lines up with on the axes, and possibly by colour or shape or something. And what we aim to do is see the variation and see past the variation. So in statistics, it's all about variation. Things vary, which is why we need statistics, and we're often trying to figure out how the variation can be explained by something else changing. So something changing causes something else to change, and we're interested in how that works. So we need to see how things change. We need to see the variation itself, but we also need to stand back and squint a bit and look at what we've got and say, I'm going to see past the variation to the overall pattern. That's what I'm looking for in graphs, both how it varies and how the pattern works through the variation. So what I'm going to do is I'm going to get you to draw a scatter plot for me. And I'm going to draw it here on the board. And it's going to go something like this. Actually, just a second. It's going to go 90. Okay, that's my axes. I have an, what's going on with the focus? I have an x-axis here. This is going to be height. These are people. I'm going to have some people. I'm going to measure their height. And I'm going to an x-axis here, and this is the weight. I don't care about your height and weight, because what I have over here is some little cards with little people on them, which list their height and their weight. So what I'd like you to do is come up here, pick a card and stick it on the graph, please. It needs to line up with, if you look at the person's weight, it'll need to line up with the weight on this side, and you look at the person's height, it'll need to line up with the height on this side. Please come and draw the graph for me, because this is way more fun. Perhaps come in this side and go out this side. No. It's got a magnet on it. Put it on the board. You're so gracious, thank you, all of you. If there's not enough for all of you, I apologise for the waste of your exertion there. Beautiful. Okay. As these people are going, you can already see there's a pattern forming, which I set up because this is pretend data, of course. But there's a pattern forming, and I can have this general noticing that if a person has a high height, they also have a high weight. On average, generally. I can see there's a bit of, you might have to shift them a bit to make them stay. Yeah. There's a bit of wiggling around there. Some of them are a bit taller than the group, and some of them are a bit shorter than the group, or a bit heavier, or a bit lighter. Hallelujah. Beautiful. Sorry. All right, that's a scatter plot. Now, one thing I need to tell you about a scatter plot is that every individual is on the graph. Look, we put them there, but each little rectangle here represents one of the people and the data about them. That's why the scatter plot is the most fundamental kind of graph. So I'm just going to draw a more traditional scatter plot. Here's a scatter plot. This data is from about the 1920s, and it's a crash test simulation. So we have some cars. That's the subjects are cars, and the cars were going along at a particular speed, and given their particular speed, then they started braking, and then it's how long it took them to stop. These speeds, I believe, are in miles per hour, and the distances are in yards, I think, so because it was in the 20s. So this is a crash test situation, and the faster a car is going, the longer it takes to stop, which is what this graph is saying. Okay, so we've got a high speed before you start stopping corresponds to a high stopping distance, and that's the general pattern. But it's not that good a relationship. There's a lot of scatter there, so it's not going to be particularly easy to predict exactly what the stopping distance is given the speed, but we can get a pretty good idea. And you see my data set up there. Each car has a speed and a distance, and that's all good. Now, the way I've drawn this graph, this one on the y-axis here, on the up axis, is called the outcome. That's what I normally call that variable. And it's in my little arrow diagram. The speed causes the stopping distance to change. So it's the outcome, also known as a dependent variable or a response variable. And this one is called an explanatory variable, also known as an independent variable. And it seems logical that the speed you're going at causes your stopping distance to change. But in statistics, it is actually impossible in statistics to prove that anything causes anything. That's what science is for. Statistics just says whether things are numerically related to each other. Numerically, a large speed corresponds to a large stopping distance. If you were a crash test investigation team, you would see the skid marks on the road, so you would know how long it took a car to stop, and you could try and calculate from that information how fast it was going. So you may want to actually do this and use the stopping distance to calculate what the speed was. So because the speed is the outcome of my calculation, it's now my dependent variable, the outcome variable. So just because the stopping distance doesn't cause the speed to be higher, doesn't mean that you can't use it as an outcome in your graph. It depends on what you're trying to achieve. So if I had the information about stopping distance, I could try and figure out the speed from it. All right, still the same relationship though, just upside down. That's a scatter plot. Scatterplot's the most fundamental graph. Each dot there represents one of the cars in my experiment. Each dot on my whiteboard represented one of the people. All the people are on the graph. A line graph, on the other hand, is actually more complex than, from a statistical standpoint, than a scatter plot, even though the line graph was the first kind of graph to ever be invented. So in a line graph, the subjects are represented on the graph multiple times. So it may be that you might measure someone, for example, you've seen those line graphs in the children's blue book that shows the progression of their heights and weights over time. That line represents a single child over time. That child's been measured multiple times. And that's how a line graph works. You put the person up on there multiple times and you connect up the dots to show that they all belong to the same person. But the information is still represented by what the point lines up with on the axes. In that one in the blue book, it's about a child's age and their weight or height, or head circumference, or whatever it is. This graph is about trees. I have 5 trees here, and I've got how many days they've been alive for, well, since they were planted anyway, and their circumference around their trunk. And each tree has a colour so that I can follow it along to see how it grows over time. And at the moment I can see that all my trees grow, they get wider over time, but they're not always in the same place relative to each other. Tree number 1 and tree number 5, for example, the red one and the pink one switch over in the middle. Okay. But the problem is, every dot on a graph inside the computer is a row in your data. That tree there tree number 1 has 4 dots on the graph. So it's going to need to be represented as 4 rows in the data for the computer to be able to cope. So currently, this is how my data would normally be set up. I would have trees, and my tree would have the circumference on day 118, the circumference on day 664, the circumference on day 1231, the circumference on day 1582. And tree number 1 would have all their data in a row, and that's how you're supposed to do it. But if I want my computer to draw a graph with the tree on it multiple times, because each row is a dot, I'm going to need multiple rows for that tree, because that tree has multiple dots on the graph. So I'm going to need to reshape my data and draw a new way of thinking about my subjects in inverted commas. My subjects are no longer trees, they are trees on particular days. And a tree today is a different subject to a tree tomorrow. They're related to each other, sure, because they're the same tree, but they're different because they're on different times. So, about a tree, I could write what its circumference is on each day. About a tree day, I could write which tree it is, which day it is, and what the circumference is. That's the 3 pieces of information about a tree day combo. And now I've got each tree is in there multiple times. So tree number 1, on that day it has circumference of 30. Tree number 1 again, on that day it has circumference of 87. Tree number 1 again, on that day it has circumference of 128. Tree number 1 again, on that day it has a circumference of 145. And then tree number 2 will have 4 rows of its own, and tree number 3 will have 4 rows of its own. This is called long data, and the other one's called wide data, just in case anyone ever mentions that to you again. But all I wanted to say is that sometimes it's useful to think about your objects as object-time combos. And so now down here, this is actually a picture of this data. I'm interested in how the day and which tree it is affects the circumference. So on my graph, I can look at a particular dot, and that tells me what the day is and what the circumference is, and the colour tells me which tree it is. And so those informations are about that row in the data. So that's a line graph. You can follow things over time. Beautiful. Coming back to my scatter plot. Now, this is great. Stopping distance is a numerical variable. Speed is a numerical variable. And weight is a numerical variable, and so is height. But what would happen if one of the variables was a categorical variable? What would I draw then, is the question. It's a rhetorical question at the moment. I'm going to get you to do it again. So stand up. I'm going to get you to sort them, draw a graph based on not height, but gender. All right, come on. It's the afternoon. I've got to get you doing something. So we're looking at the weight and the gender. If you look closely, you might notice these people don't weigh very much given their height. It's because they're stick figures. So bad, I know. All right, so how are you going to... Yeah, that's a good plan. Right, so already some people are using something really interesting. They're having to solve the problem of having to put people on top of each other, which means you're spreading them out a bit. That's a good plan. So just move them over to the left or the right so that you can fit them in. Might as well just put the rest of them on. Okay, so there's my graph there. I had to spread the people out a bit side to side so that I could fit them all in, because even if my magnets were strong enough, if I put them on top of each other, it might not be as obvious as what's going on there. And I can see that my men, sort of on average, are a little bit heavier than the women, which is what you'd normally expect, but it is made-up data, of course. So there we go. And I can also see that actually there is a lot more men up here than there are men down here because of that wide cluster there. Okay. So that's my second kind of graph. Okay, I have a categorical explanatory variable and a numerical outcome variable. This particular graph is about guinea pigs. Guinea pigs were given a vitamin C supplement daily. They either had a vitamin C tablet, which is VC, or orange juice. And they got a low dose or a high dose, and then after a certain amount of time we measured how long their teeth were, because of course vitamin C is good for gum health and therefore tooth health. Okay, so this is what we have, and that here, there's a problem with this graph in that some of my guinea pigs are on top of each other. Look at number 7 and 8 here. It's got low for the vitamin C dose. They both have low for the vitamin C dose. They both have 11.2. So this dot just here, about there, is 2 guinea pigs on top of each other. So what we're going to do is we're going to spread them out a little bit, and this is called, it's got a name, it's called jittering. This is a jittered scatterplot, in case you want to know what it's called. So that's a jittered scatterplot. And jittered scatterplots are totally awesome and are really the best graph of all time. Every guinea pig is on the graph. I just had to move it slightly sideways so that I could see what it is. It still lines up with the correct thing on the y-axis, and because it's near the low, above the low one, we put it in the right spot. So that's great. I can see that if you have a high dose of vitamin C, you get a longer tooth length. I can also add some colour to figure out how the vitamin C supplement makes a difference. So I'm going to add in the colour, which is supplement. So the orange is orange juice and the blue is vitamin C tablets. And already you can see that if you have orange juice, it's higher than it is when you have vitamin C tablets, because the red dots, orange dots tend to be above the green-blue dots. So we can add in more relationships by changing the colour or shape of the dots as well. So there you go. All right. So now we have what happens if they're both categorical. I'm not going to get you to do that one on the board. What if both of the variables are categorical? I could have blonde, dark and red for hair colour and brown and blue, hazel, green for eye colour. This is a class of statistics students in about the 60s, I believe. And I could give everybody, I could line people up with what their hair colour is and what their eye colour is, but of course if I did that I'd just get 6 dots because everybody's on top of each other. So I just jitter it in both directions and I get this. Now I can see that if you have blonde hair, it's very unlikely to have brown eyes, because there's a heap down here under blue, hazel, green and a heap up there. If you have dark hair, then it looks like it's roughly 50-50. If you have red hair, it looks like it's ever so slightly more likely to have blue, hazel, green, but not particularly. And if you have brown eyes, you're very likely to have dark hair, if you look along the top column here. So now I can see the relationship between things. You will not see this graph very often in a journal article, but that's just because people really have not discovered the wonder of the jittered scatterplot for categorical relationships. But I will get on to the sort of graph that you will normally see later. And I could add in colour and see if gender makes a difference, and it doesn't appear to make much of a difference. I've coloured in the males that orangey colour and the females that greeny blue colour. Okay, one more scatterplot. What if I've only got one variable? What if I'm not relating it to anything and I'm just going, hey, what can I know about how long it takes for the old faithful geyser in Yellowstone National Park to do its eruption. There's a geyser, it shoots superheated water up into the air every few minutes, and this is how long it takes to do that. And I can just put a dot that lines up with the time taken. I don't have a second variable, that's okay. I can already see that it's really unlikely to have a time between 2.5 and 3 seconds because there's a gap just here. But I can't quite see how well clustered these is, so I can jitter it a bit. And I can actually see there's a really good cluster just here between 1.5 and 2 seconds. You won't see this graph very often, but the reason I've done it here is so that I can show you about the two graphs that you will see very often. A histogram shows you how much data is in each zone. So I will take my geyser eruption times, and I will have a look at the spread that they have, and then each I'll cut it into equally sized pieces, and then I'll draw a bar which says how much data is in that zone. So in this first zone, there's quite a bit of data, and so it has a high one. This one over here, there's only 1, 2, 3, 4, 5 data points, so it only goes up to 5. So I've really counted how many dots are in that zone and drawn a bar to represent how high that is. And your histogram would normally not have the dots on it. That's what a histogram is. It counts how much data is in each zone. And so instead of jittering it and having a look at how closely clustered the dots are, I can just look at how high the bars are to see how likely it is to be in any particular zone. And it's most likely to be here between 4 and 4.5 seconds. A box plot, on the other hand, shows how spread out each quarter of the data is. So here's my scatter plot. What I'm going to do is I'm going to draw some lines that divide that data into quarters. So I'm going to start in from the left, and I'm going to move in from this side, and once I get to a quarter of the dots, I'm going to draw a line, and I'm going to move along, and once I get to half the dots, I'll go draw a line, and once I get to 3/4 of the dot, I'll draw a line, and then I'll draw one at the end. And that's what a box plot is. Okay? This zone here contains a quarter of the dots. This zone here contains a quarter of the dots. This is a quarter, this is a quarter. So I can see that that second quarter there is more spread out than the other quarters. That's what a box plot is for. And normally you wouldn't see the dots on a box plot either. So back when I was drawing that jittered scatter plot for my categorical numerical relationship, or here on the board with the male and female, you'll often see two box plots next to each other. You'll convert this dots into box plots and these dots here into a box plot, and you'll see two box plots lined up next to each other. The problem with a box plot, of course, and a histogram, oh yeah, just a second, before I get onto that, these markers have names just in case you're interested. I'm not going to test you on it, but I just thought you should know. So the middle there, the halfway mark, where half the data is below and half the data is above, that's called the median. The minimum is the smallest one, the maximum is the biggest one. The quarter mark is called Q1, and it's the first quartile. And the 3/4 mark is called Q3, and it's the third quartile. Technically, the median's Q2. It's the second quartile. So, and the space between Q1 and Q3 is called the interquartile range. So if you ever see in a medical journal they talk about an IQR, that's what they mean, the space between the two quartiles. So what that means is that the middle half of the data is that wide. Because there's a quarter in each of these zones, that box contains half the data. So the middle half of the data is that wide, and it's a measure of how spread out the data is. Now here's the thing. The individual subjects are not on these graphs. So the scatter plot, each dot represented one of the people or guinea pigs or whatever it was in my study, but the box plot doesn't have any of the actual individual objects in my study on it. Those numbers that are in the box plot don't belong to any one guinea pig or geyser eruption, they belong to the whole set of data. And that height of that bar there doesn't belong to any one piece of data, it belongs to the whole set. So these graphs are very interesting from a sort of a philosophical point of view, because they actually display information about the whole set of data as opposed to about each individual object. So each individual person on the scatter plot, you can find out what their height and weight is, but if I drew something like a Boxplot or something, I wouldn't be able to tell what the individual people were, but I could just see the whole group. And that is my next point. Variables don't have to be recorded about individuals, they can be recorded about a group. You might have a group of 10, 1, 2, 3, 4, 5, 6, 8 individuals, and you might record, instead of just recording information about each person, I can combine that information to create other things. So that group of people, I could write down the median for the whole group. I could write down the mean for the whole group. I could write down the percentage of the temperatures that are above 37 degrees. I could write down the IQR of the temperatures. I could write down the standard deviation of the temperatures, which is another measure of spread. I could write down the maximum temperature. All of those bits of information don't belong to any one person, they belong to the whole group of people. By the way, these are called measures of centre, and these are called measures of spread. So the centre ones tell you roughly, you know, the whole group, where's the middle of that group? That's what the median and the mean are for. And the IQR and standard deviation are about how spread out the data is. Okay. So here's the thing. What if I wanted to draw a graph about my groups as opposed to about my individuals? Well then, if I wanted a dot on my graph to represent a whole group, then I would need a row in my data for the whole group, because that's how computers work. Each row is one of the dots on the graph. So now I have a whole group of people, instead of each individual person with their chili and their temperature, now I have a whole group, and I've written down that whole group had chili and their mean temperature was 37 and the percentage of that were above 37 was 50%. And the second group has its own row, and it's a nice little data set. This is called aggregate or aggregated data. And that's the sort of thing that you would show when you did your research report. If you did one, you wouldn't show everybody's data, you would just show the group data. And that's called aggregate data. And I can draw a graph of aggregate data. So here's my guinea pigs. So instead of thinking about individual guinea pigs, I could think about groups of guinea pigs. So I have a group of 10 guinea pigs, and about the whole group I could write down what dose of vitamin C they had, and about the whole group I could write down what kind of supplement they had, and about the whole group I could write down the mean tooth length. And those are the dots that represent these things here. So this dot down here, blue means they had vitamin C tablets, they're on the low dose, and they got about 7.98 for their mean of that whole group of 10 guinea pigs. So I've replaced that group of 10 guinea pigs with a single dot. But I don't just have to record the mean on my graph, I could also record the standard deviation, and I could put some error bars on. I could go up from that point a certain amount and down from that point a certain amount by figuring out the standard deviation. But that's a little difficult to read, so I'll just jitter it a bit and move them over. And this is the sort of graph that you might actually see in a research paper. Normally you'd actually have the colours connected with lines as well. In my head it doesn't make any sense because that's not the same group twice, it's two different groups, so I don't draw them and connect them with lines, but people do all sorts of things that don't make sense to me, so that's fine. So that's the sort of thing you'd normally see. Each dot there is a whole group of guinea pigs, and the height above and below is the standard deviation. You could also draw a box plot for each of them if you wanted. That's group statistics. So I've stripped away all the individual guinea pigs and now I've just got the groups so that I can see on average, the same as what I saw before, that if you have orange juice you're higher, if you have a high dose you're higher. Well, let's look at this one. Yes?
So when you're saying this, the standard deviation, you're saying like 95% confidence. For standard deviation, you're saying a single or like...
Well, I mean, this one's one standard deviation, which would mean that you'd expect 2/3 of the data to be between the top and the bottom. 95% confidence interval we will get into, but you would expect 95% of the data between top and the bottom. Yes, that's a worthy point for the recording. In lots of... people have a choice as to what their error bars represent. They may represent standard deviations, they may represent standard errors, which is a whole different thing, and I'll get to that in another lecture. They may represent confidence intervals, and any paper that doesn't tell you which one is which is missing some very important information, because you need to know what that represents about the group. Okay, at the moment that represents how about 2/3 of the guinea pigs you would expect to be in that zone. Right, here's my hair color, eye color graph, I could supposedly, for each group of hair color, split my people into 3 groups of hair color and write down the percentage that had blue, green, hazel eyes, and I could draw those as dots if I wanted. This is not the traditional way to represent this. Normally you draw it as a bar, like that. Because it feels like a histogram, because you're counting how many dots there are in a particular zone. Okay, so there I am. I have almost 100% of the blonde people have blue eyes. About half the dark-haired people have blue eyes, and a little bit over half the red-haired people have blue eyes, which is what I noticed earlier. That was my good suggestion, but this is being more accurate so I can tell the difference. You can put the brown eyes in too. You could put them on top. That would be a stacked bar chart. You could put the brown ones next to it as well and have a column of blues and a column of browns. It doesn't really matter. Most of the time you'll only put one of them, whichever one you're most interested in. If there's only 2 options, you usually only put one. So that's a bar chart. So in general, if you want to talk about a particular numerical value, like an average, you put dots. And if you want to talk about a percentage, you'll put bars. However, in most medical journals, they'll put bars for means as well. So back on this graph up here, you'll often see a bar that goes from the bottom up to where that dot is to represent the mean. But I feel uncomfortable with that because bars always feel percentages in my head, and so I have trouble reading those. Okay, so that's graphs of group statistics. The point being that I have removed all the individuals and just drawn information about the groups. So when you read a graph, decide to yourself, are the individuals on the graph or is it showing me about the whole groups? Now I've only got, that clock is crap, I've only got a couple of minutes to go, and so I'm going to do the last bit kind of quick. The biggest idea in statistics is also about graphs, and I'm going to come back to it next week. The biggest idea in statistics is that your subjects, your bananas or your guinea pigs or your geyser eruptions or your cars, are just some of the subjects that you could have had. You could have had other cars or other guinea pigs. Your group of subjects is just one of the groups you could have had. You know, I've got 40 guinea pigs. Well, I could just as easily have had a different 40 guinea pigs or a different 40 guinea pigs. There are lots of groups that I could have had. So therefore, any one value of a variable is just one of the values that I could have had. So I've got a height here of 162.8. That's just one height that I could have had. And even if I look at all of these heights, well, it could still have been some other height that isn't represented there. Just one of the things that I could have had. Now, that's even true if it was calculated from other variables, and it's even true if it was recorded on the whole group. My guinea pigs today had a mean of 7.98, but tomorrow, on a different group of guinea pigs, maybe it would be a different number. Okay, here we go. So we're getting close to the biggest idea in statistics. So any one value of anything that I could possibly write down or calculate on an individual or a group could have been something else. The description of all the values that could have been and how likely they are is called the distribution. That's a statistical terminology. The distribution of the variable describes all the things that could have happened and how likely they all were. Okay. The biggest idea in statistics, we're nearly there. It has 3 parts. Everything could have been something else. The description of all the things it could have been is the distribution. And the biggest idea in statistics is that everything has a distribution. Somewhere in the universe, somewhere in the fabric of the universe, there is a description of everything that could have happened and how likely it was. Now, when we think of rolling a die, you know what that is. It's like 1/6 for all the options. But some things are more complicated than that. Doesn't matter. So if you choose your variables right and the way that you measure things, you might even know what the distribution is called. It might be such a commonly occurring distribution that it has a name, which means it'll be easy to do some calculations based on it. That's the big idea in statistics. If you can think, what are the values it could have been, and think about how the probabilities are distributed, then you have a chance of answering some of the questions that you want to answer. All right. So just to point out, because this is about graphs, graphs of test distributions tend to look like sort of smooth histograms. So the height there tells you how likely it is to be near that spot. So just like a histogram, the height told you how likely it was to be in a particular zone. Same deal here, only the zones are really tiny. So an individual guinea pig, its tooth length might look like this. That's called a normal distribution. It's really likely to be near 20 and not so likely to be near 30 or 10. Oh, it's called a probability density function, by the way. That's its official name, shortened to PDF, which is confusing. But there you go. I might take individual guinea pigs, and each individual guinea pig, that's how likely the things are that they could be. But because with 10 guinea pigs, they'll all be different, when I do my averages, it turns out to have its distribution of its own, which is also normal, but skinnier. And it doesn't matter what I measure, even if it's a standard deviation, it will have a distribution. The standard deviation's distribution looks like that, not normal. But, you know, still something that has a name. It's called the chi-squared distribution, if you're interested. And they're not named by any particular useful naming system. I wish they were, but they're not. So there you go. So that's the concept there, is that anything that I could possibly write down or measure about an individual or a group has a graph like that to describe how likely all of the things that could have been are. And we'll get on to more of that next week. That's pretty much everything I wanted to say about graphs. And I'll move on from those distributions to talk about hypothesis tests and confidence intervals next time. Thank you.